Good morning and welcome to our annual community engagement event. My name is Ruth Kruger and I'm the strategist for knowledge translation and communications for the Ontario Neurodegenerative Disease Research Initiative, or ONDRI. This annual event that we host was previously known as Friends and Family Day, and it's meant to involve all our study participants, their families, care partners, as well as our wider community of stakeholders in our ongoing research. So we invite you to listen to our speakers, ask questions, and participate with us today. A couple housekeeping notes first. You can see the session is being recorded. We will share a link with the recording when it's ready. Also, we've muted you all so as to have no noise interference, which can make it hard to hear what's going on. But feel free to ask questions by clicking on the chat box and we'll answer them following each speaker's presentation and throughout the event. Our leads, Dr. Mario Macelis, Dr. Doug Munoz, and Dr. Richard Swartz, along with many other esteemed researchers, colleagues, and supporting team here at ONDRI, have been doing this important work for many years now. Throughout this period, we've been generously supported by the Ontario Brain Institute and guided by our Patient and Community Advisory Committee, or the PCAC. ONDRI's research has made significant progress in unraveling the complexity of neurodegeneration and dementia. And to that end, we have an exciting agenda for you today. So let's start. Our first presenter, Dr. Mario Macelis, will take a few steps back and provide some of the big picture behind the research by addressing questions such as, why is diagnosing dementia and its many causes so complex? What do we know? And what are some of the basics we're focusing on in our research? He'll then tell you a little bit more about ONDRI, where we've been and where we're going. Next, we will hear from Dr. Doug Munoz. Doug will start with a general talk about the biomarkers or early indicators of these diseases. He will then introduce the different types of biomarkers and focus on blood-based biomarkers and the work being done in this area. We will then move on to our work in remote monitoring and wearables. Dr. William, or Bill McElroy, our lead in this area, will talk about all the background work that his team has done and how this work is leading to potential new biomarkers in activity, sleep, speech and language, and other areas. He'll also expound on the health and activity reports the AIM2 team has developed as part of the HANDS Ontario research study and its predecessors. Bill's talk will be broken into two parts to give you a well-deserved break at approximately 10:30. Next, we'll feature Dr. Susan Bronskill and Laura McLagan of our AIM4 Health Services research team. Susan and Laura will explain how health administrative data expands out to look at the population and what we can learn from these data in terms of population health and implications on people we study through ONDRI. We will then introduce our HANDS Ontario study that's currently taking place. Dr. Richard Swartz will introduce HANDS and the objectives and framework for this research. We will hear from Larry Linton, an important member of our PCAC. Larry will tell us a little bit about living his best life with Parkinson's disease and about his experience participating in the HANDS Ontario study. We will then wrap up on HANDS Ontario and answer any remaining questions on our research. Now I have the privilege of introducing our first speaker, Dr. Mario Macelis. Dr. Macelis obtained his MSc in pharmacology in 1997, his MD in 2001, and his board certification as a neurologist in 2006, all at the University of Toronto. He completed a clinical fellowship in cognitive and movement disorders in 2008 and obtained a PhD in clinical neuroscience in 2012. Dr. Macelis is an associate professor of medicine at the University of Toronto and a clinician scientist at Sunnybrook Health Science Centers. His research focuses on understanding shared genetic risk for neurodegenerative disease of aging using genomic, cognitive, and neuroimaging data. He is, of course, also the co-lead of the ONDRI study. Welcome, Dr. Macelis.
So welcome, everyone. So I'll first start by introducing a little bit about dementia and what it is. There's a crisis brewing, the dementia crisis, and a tsunami is coming our way. And dementia is a leading cause of dependency and disability among seniors and can have a significant impact on social, mental, and physical well-being for patients and caregivers. I think that most people who are attending today has, in one way or another, knows someone who has dementia or family members who are providing care for those with dementia. It's touched 2 out of every 5 people. 1 in 10 Ontarians over the age of 65 have dementia. 1 in 5 Ontarians have cared for a person with dementia, and 1 in 3 seniors die having some form of dementia. So this is a real crisis that needs to be dealt with urgently. Dementia will affect an increasing proportion of the population, and this is really due to the growing age of our population and the aging of the baby boomers in particular. And this will ultimately result in an increased incidence of dementia. The strongest risk factor for dementia is not genetics, is not environmental factors, is not cardiovascular risk factors. In fact, the strongest is aging. So as people age, dementia is going to become more prevalent. And what this figure here is showing is that if you look at the current statistics, the majority of our population is kind of between the ages of about 40 and about 65. But in the next several years, by 2036, you're going to see a shift where the majority of the population is going to be in between the ages of 70 to 90. And as a result, dementia is going to be imposing a greater burden on our society. The risk for dementia doubles every 5 years after age 65 to about 25% by the age of 85. And about a quarter of Ontarians will be expected to will be greater than the age of 65 by 2038. This is going to become an increasing burden that needs to be addressed. What is dementia? So it's an umbrella term that is used to describe a progressively worsening syndrome. A syndrome is a group of signs and symptoms that someone suffers from that occur together and produce a clinical condition. So dementia is a progressively worsening syndrome characterized by problems with memory, decision-making, speaking, and behaviors that ultimately result in difficulty with day-to-day tasks and a loss of function. And the key here is that it's progressive. So dementia will generally always worsen. And there are many different causes of dementia. Neurodegenerative diseases are the main cause, but these can also co-occur with cerebrovascular disease or stroke. And in fact, over the age of 65, even though in textbooks, Alzheimer's disease is listed as the most common cause of dementia, in fact, over the age of 65, most commonly it's mixed diseases. So it's usually a combination of Alzheimer's disease combined with small vessel disease of the brain or Lewy body dementia that are the most prevalent forms of dementia. So as you can see here, based on this image, neurodegenerative diseases are the strongest risk factor for the development of dementia. However, what we see in life when someone comes into my clinic and I diagnose someone with Alzheimer's disease, we don't always get it right. And I'll be going into more details on this in the next slide. So the only way of definitively confirming if someone has Alzheimer's changes in the brain is currently on autopsy or pathology. After someone has passed away with one of these conditions, we're able to do an analysis on the person's brain and be able to confirm what the underlying changes in the brain are that are actually contributing to their clinical syndrome of dementia. So the concept here is that dementia is diagnosed in life by doctors and physicians, and we have a certain degree of ability to get the diagnosis right in life as to what the underlying causes are. However, we don't always get it right, and what we can't predict right now are those that have mixed pathologies or mixed causes in the brains. So what this slide is showing here are the neurodegenerative pathologies or proteinopathies which cause dementia, and these are the most common causes of dementia. And when I talk about proteinopathies, almost all dementias are caused by the accumulation of proteins in the brain, and proteins are the building blocks of our cells. And with the accumulation of these proteins in the brain in an abnormal way, because these proteins normally help the brain function properly, what happens is there's neurodegeneration or loss of brain cells or neurons. And so these are the most common pathologies. Here, I show you the typical plaques, amyloid plaques, and tau neurofibrillary tangles, which cause Alzheimer's disease. So this is the pathology underlying Alzheimer's disease. Next to it, I show you alpha-synuclein inclusions in the form of what we call Lewy bodies in the brain. And this is the main cause of Parkinson's disease with mild cognitive impairment, Parkinson's disease with dementia, and also dementia with Lewy bodies. And this is the second most common neurodegenerative cause of dementia. In the slides below, we show the different causes of frontotemporal dementia, or FTLD, which is frontotemporal lobar degeneration. And there are two major causes, the tauopathies, and tau is a protein that goes awry in this disease, in these groups of diseases. And then the TDP-43 proteinopathies, which not only can cause frontotemporal lobar degeneration or frontotemporal dementias, but it can also produce ALS as one of the most prevalent causes. So these are the major pathologies that we see as causes of dementia. And just to remind you, if you're over the age of 65, you're more than likely to have two or more of these pathologies in combination rather than one. We also must not forget cerebrovascular disease, or what we call cerebral small vessel disease, which can co-occur with all of these neurodegenerative pathologies and thus alter and change the progression rate of dementia and the way it presents. So dementia overall is extremely complex, and it's difficult to diagnose because of this complexity. And as I had alluded to, the clinical diagnosis does not always match what the pathology is. So in other words, if we have someone who is clinically diagnosed with Alzheimer's disease, they may have Alzheimer's pathology, but they may have other pathologies that we were not able to predict. Unless we follow patients over time, it may become more evident that there are other contributing diseases. The other thing that's very helpful in diagnosing these other contributing diseases are modalities such as brain imaging. So if we do an MRI scan, we can see hardening of the arteries in the brain in the form of small vessel disease and know that someone may have small vessel disease combined in. So we need to do research that helps us understand the relationships between the different stages and severities of the disease, because right now they're not well understood or associated in a linear fashion. So the ANDRI study wants to study these clinical syndromes, understand more about the underlying pathology that causes these clinical syndromes. And there's been a shift. We are starting to focus on biomarkers, and I'll go into a little bit more about this, and Dr. Munoz is going to go into a lot more detail about this. But biomarkers, the hope of using biomarkers is to be able to better refine our clinical diagnosis to be able to identify those that have pure forms of dementia, such as pure Alzheimer's disease, from mixed forms, such as Alzheimer's mixed with Lewy body disease or Alzheimer's mixed with small vessel disease. Ultimately, by understanding this complexity here, we want to understand how dementia functionally impacts patients and their families, as well as their free living behavior, such as sleep, exercise, social interactions. And this is another biomarker that Dr. McElroy is kindly going to be presenting on a little bit later on. So I alluded to biomarkers, and now I'm going to give you a definition of what biomarkers are. So biomarkers are needed to unravel the complexity of dementia, because if we just see someone in our office, we're not 100% certain whether or not they have Alzheimer's disease or Parkinson's disease, or mixed forms of the disease. So we need biomarkers, which are characteristics that can be used to objectively measure and evaluate an indicator of normal biological processes or pathogenic processes. And what I mean by that are the underlying changes I was alluding to in the brain. And ultimately, biomarkers may be used to monitor response to new therapeutic interventions that are being developed. So what else are biomarkers used for? So we want to be able to identify people who are at risk of developing disease, so disease susceptibility, or a trait that they may possess that puts them at a higher risk of developing a disease like Alzheimer's disease and developing dementia in general. we want to be able to use these biomarkers in the prodromal stages, so the stages before the full disease manifest, where, as someone's aging, they may have some very minor symptoms that may be just attributed to aging, but may also be attributed to pathological or abnormal aging. And so we want to be able to use biomarkers to determine if this is just kind of a change related with the normal aging process, or whether or not there may be an underlying pathology contributing to it. In someone who has full-blown symptoms of the disease, we want to be able to add biomarkers to the clinical diagnosis to state with more certainty whether they have Alzheimer's disease, whether they have mixed forms of dementia. We want to use this to help refine our differential diagnosis. We want to use biomarkers to track the rate of disease progression to identify people who will be more likely to decline rapidly versus people who have a more slower, indolent process. And then ultimately, when we have the holy grail available, that is disease-modifying therapeutics, we need to use biomarkers to track response. Ultimately, by adding biomarkers into the Andri program, what we're hoping to achieve here is personalized or precision medicine. So we want to be able to look at an individual and to be able to define what the underlying contributing factors are to their clinical neurodegenerative dementia syndrome in order to be able to better guide and target therapies that are hopefully coming down the pipeline. So these are some examples of biomarkers that are already available in Alzheimer's disease. Unfortunately, they're not readily available in the Ontario healthcare system. But one biomarker is amyloid PET. And if you look at an individual who does not have Alzheimer's disease and compare it to an individual who does have Alzheimer's disease, and you use a special radioactive ligand that's injected into your veins and taken up by the brain, what we see here is that Alzheimer's disease have this kind of fluffy enhancement of their brain imaging, their PET imaging pattern, and that indicates that they have Alzheimer's pathology. If you look at structural MRI, so this is the sequence that we get in individuals who are participated in the Andri 1 program. What you can see here is that this is a control brain, and you can see there's very little shrinkage of the brain. But on this slide, you can see there's a lot more black, and the black is the space around the brain and in the center of the brain. And this represents atrophy or loss of brain cells or death of brain cells. And so when we see shrinkage of the brain, that's indicative of significant neurodegeneration, and different patterns of shrinkage are suggestive of different neurodegenerative diseases. Finally, there's new work being done with other PET imaging tracers. These are tracers that can highlight different proteins in the brain. And now we have tau PET available in the research setting that can indicate if someone has the tau conformation that's typical of Alzheimer's disease. We also have available biomarkers in the cerebrospinal fluid. So this is taking a sample of spinal fluid from someone's back, and we can send it to labs to determine if they have amyloid and tau pathology indicative of amyloid changes in the brain. And Dr. Munoz is going to be talking a little bit more about this. So there's an urgent need for disease-modifying therapies targeting these neurodegenerative pathologies, and we need biomarkers in order to be able to define what the underlying pathologies are in individuals who suffer from dementia. But as you probably are all aware, essentially all disease-modifying therapies have failed the gold standard of what we call the clinical trial, where we randomize a group of people to receive a drug, and we randomize a group of people to a placebo, which is essentially, a salt injection or a sugar injection or a sugar pill. And what we hope to show with these clinical trials is whether or not the disease group does better than the placebo group. Now, essentially, all clinical trials of Alzheimer's disease have failed, and I know that people have heard about the aducanumab story, which also has been a very controversial decision to approve in the US by the FDA, because most clinicians, when we look at the data, it doesn't seem to benefit patients. It may show some changes in biomarkers in the brain, but it doesn't seem to benefit the patients. Ultimately, what we want to be able to do is to figure out why these clinical trials have failed. And one reason why these clinical trials may have failed is because in life, we were not able to predict the specific pathologies that were causing the dementia syndrome. And as a result, even if we gave someone an amyloid drug, we didn't address their tau pathology, we didn't address their Lewy body pathology, or we didn't modify their cardiovascular risk factors, which caused, silent strokes in the brain. Ultimately, we want to use biomarkers to assist with defining outcome measures. We want to identify slow from fast progressors. Another reason why clinical trials have failed is that they've been given to people who are in the later stages of the disease. And once neurodegeneration, once there's been significant loss of brain cells in a person suffering from dementia or any neurodegenerative disease for that matter, it's probably too late for any of these drugs to work, even if they do appear to remove amyloid or to remove tau from the brain, for example. So we need to understand more about the basic disease mechanisms. We need to understand genetic risk. We need to understand and address other risk factors such as cardiovascular risk, so hypertension, diabetes, high cholesterol, smoking. These are all ways we can intervene to reduce the risk of developing dementia. So what do we hope to achieve in ANDRI? So the mission statement of ANDRI is that we're devoted to improving the diagnosis, the prognosis, the care, and outcomes for people living with neurodegenerative diseases, such as stroke and dementia. There are several different aims that we have. The first aim is to take advantage and analyze all of the data that we collected during the first phase of ANDRI, and I'll talk a little bit about that in the next slide. The second aim is to look at wearable devices that can be worn by patients at home to help track how people are doing with the disease, help to track their activity, their sleep, and to assess health and function in a person's home and community. And an extension of this is the HANDS Ontario program, which will be discussed a little bit later. We want to not only work in isolation. In order for us to overcome all of the challenges and complexities of diagnosis, we need to collaborate. We need to bring our ANDRI data set into the space of other large dementia data sets, large neurodegenerative disease studies, in order to combine our efforts, to increase our sample sizes, to combine our data in order to achieve what we call higher statistical power, which allows us to better easily detect small signals in large samples, which allows us to be able to understand more about individual risk for disease, understand a bit about the basic science underlying this and how this applies to the health system, which relates to aim 4, being able to harness the power of data that's collected across Ontario at hospitals, through pharmacy records, by analyzing health system records. And this allows us to track progression of disease. And so Laura and Dr. Bronskill will be discussing a little bit about this and available to talk about this later. So where were we in ANDRI? So I'll first talk about the foundational study. We studied 520 participants with a variety of neurodegenerative diseases, so cerebral vascular disease, frontotemporal dementia, Parkinson's disease, with and without cognitive impairment, Alzheimer's and mild cognitive impairment, and ALS. They were followed over 3 years, studying many different platforms, so a clinical platform, genetics, cognitive testing, gait and balance assessment, eye tracking, brain imaging, retinal imaging, and we've added a neuropathology and biomarker core, which is a really exciting add-on to this original diagnosis. And now this ANDRI data is not only available for us to study, but it's available for researchers around the world to study. And this is an exciting breakthrough, and we hope to have many more publications coming out from this foundational cohort. So right now, we are only seeing the tip of the iceberg, which is the clinical diagnosis of dementia. And what we want to do is use all of these powerful biomarkers that are in development and some that are proven to be able to resolve the complexity underlying dementia, to better understand these diseases at the microscopic level in living humans. And so this is why we want to, we're collecting autopsies in patients who consent to provide them, so that we can understand the pathology that causes their dementia syndrome. And in life, we're adding the genetics and molecular platform. So I'll stop there. So the next speaker is Dr. Doug Munoz. He's a professor in the Center for Neurosciences Studies and Departments of Biomedical and Molecular Sciences, Psychology, and Medicine at Queen's University. He obtained his PhD in neurology and neurosurgery at McGill University, followed by a postdoctoral fellowship at McGill University and subsequently the National Eye Institute at the NIH. Dr. Munoz was recruited to Queen's University in 1992. The main goals of his research are devoted to understanding the neural circuitry controlling eye movements and using knowledge of the circuitry to probe a variety of neurological and psychiatric disorders. He currently holds a Tier 1 Canada Research Chair and is co-lead of the ANDRI study. So welcome, Doug.
Thank you. Good morning, everybody. And it's a great opportunity for me to be able to talk to you today and to continue on this theme that Mario introduced about the search for biomarkers and the search for early biomarkers of disease, so that we can hopefully intercept the disease trajectory earlier and ultimately be able to slow it down or stop it. And I'm going to focus my presentation this morning on the hunt for a blood test to diagnose neurodegenerative diseases, but I'll also show a little bit of data of other kind of biomarkers of neurodegenerative disease and the work that we're doing within ANDRI to hunt for those biomarkers. And so Mario nicely introduced the foundational study of ANDRI. And just cleaning up my desktop here. And the different disease cohorts that went in: cerebrovascular disease, frontotemporal dementia, Parkinson's disease, Alzheimer's disease, mild cognitive impairment, and amyotrophic lateral sclerosis. 520 patients, and they were followed longitudinally. And as Mario introduced, there were a number of important assessment platforms, a full clinical workup for all the patients, a blood test, a blood draw that was taken so that we could do genetics and genomics. And I'm going to come back to those blood samples later in my presentation. And then assessments of a number of behaviors, neuropsychological performance, gait and balance, eye tracking, and then imaging, brain imaging, and retinal imaging. And all of these data were collected from these patients. So we had 520 participants enrolled, and really the hunt was on for early biomarkers across all of these platforms and trying to identify where we could have the most reliable measures of these different forms of disease that Mario introduced. So I'm going to go way down in the brain, down to the molecules, to talk a little bit about the start of the disease process. So this is a schematic from deep in the brain, and this red membrane here is to be representing the surface membrane of a brain cell. And so in dark down here, this is inside the brain cell, and up here is outside the brain cell. And in all the cells we have in our brain, they're very complicated with these different proteins that might stick through the membrane, connecting from the inside to the outside. And this highlights just one of these proteins called APP that has this transmembrane domain here. And there are enzymes that are going to come along and cleave this protein regularly through life and break it into small components that are called the oligomers. And then this is now circulating in the cerebrospinal fluid in your brain. And when you go to sleep, these kind of molecules, which are like small toxins almost, they're waste products, get flushed out of your brain. But in some individuals, and for some reasons that we don't know, these oligomers, which are sticky, can start to clump together. And they make these fibrils, and then these clumps get bigger and bigger, and eventually form these large plaques. And many of you have probably heard about plaques in Alzheimer's disease. And back 30 years ago or so, when I was a graduate student, this was believed to be the cause of Alzheimer's disease. However, we learned a lot more about the science along the way. And so this is this same pathway of the oligomers forming plaques. But these oligomers also circulate through the cerebrospinal fluid and can start to attack other brain cells and start to damage the synapses that are on the outer processes of these other brain cells. And so this schematic now shows how this might look if we had a healthy brain cell shown more in red here, and an unhealthy brain cell, a diseased neuron, because various factors have made this neuron, this brain cell, prone to dying, including the attack of those oligomers. And now if we look inside one of these processes of this diseased neuron, and we compare it to a process of the healthy neuron, we start to see problems. So in the healthy neuron, there's a network of what are called these microtubules that are used to move various molecules and vesicles up and down from the cell. So material needs to move from the center of the cell out to the periphery and vice versa. And this network of microtubules is like the highway that those molecules are moving on. And as these neurons get ill, these highways, these microtubules start to break down, and the proteins here start to be released, including tau. And the tau here, shown in blue, can become hyperphosphorylated so that now it starts to clump together, kind of like the oligomers did outside the cell. Now the tau is clumping together inside the cell to make a tangle that's going to make this cell start to die. Here's another molecule in the brain called neurofilament light. This is a brain cell. It has a process which is called an axon, which is going out to create synapses, and these are the connections to new brain cells. So this axon is how the information's being propagated. And if we look inside the axon, we can see these other molecules called neurofilaments providing this long scaffolding structure in the axon. And this neurofilament light, if the axon is damaged in some kind of way, this neurofilament light can start to break down and then circulate within the cerebrospinal fluid at some concentration. And evidence of this neurofilament light means that there are sick and dying brain cells. And what's interesting is some of these markers can manage to get out into the blood. And I'll come back to that later. And so neurofilament light is another one of these potential markers that appears in multiple neurological diseases, including the ones we study in Andre, like Alzheimer's and dementia, stroke, amyotrophic lateral sclerosis, and Parkinson's disease. And so in Alzheimer's disease, some of these markers like A-beta oligomers go down. As I mentioned, tau levels in the CSF go up. Neurofilament light increases in the CSF. So these are potential biomarkers to tell us who might have Alzheimer's disease or be leading to getting Alzheimer's disease. And I literally cut this slide from Dr. Maselli's talk 20 minutes ago and inserted it in my own because I didn't have this link. And he talked to you about these proteinopathies. And so I showed you beta amyloid and tau, which are the proteinopathies for Alzheimer's disease. And in the other neurodegenerative diseases, similar problems are happening, but it's a different protein that's under having problems. So for example, in Parkinson's disease, as Dr. Masella said, it was the synucleinopathies. It's TDP43 within frontal temporal lobar dementia and ALS. And there are other tauopathies in frontal temporal dementia as well. And so there are a number of different proteins that are going awry, the coding of those proteins in the brain, and these are all producing potential biomarkers in the cerebrospinal fluid if we can figure out how to measure them. So this is a wonderful schematic diagram now that's about 10 years old that sort of shows the progression towards Alzheimer's disease. So on the horizontal axis, this is time. And I put two markings here. The time when an individual would go from being preclinical to a neurologist or geriatrician might give them a diagnosis of mild cognitive impairment. And then this second line would be when the clinician would switch the diagnosis now to Alzheimer's or dementia. And if we look at the pattern of changes in the brain over time, before and after these diagnostic signposts, there's important things I want to show you here. So first of all, when we look at the molecules in the cerebrospinal fluid that are going abnormal, the beta oligomers, tau, those molecules are changing and their levels are increasing well in advance of any clinical diagnosis. This, we believe, is the earliest part of the disease process. And it could be happening 20 or 30 years before the clinical diagnosis. And those proteins that are accumulating and those abnormalities are starting to attack neurons. They're leading to sick brain cells and synapses that are disintegrating. And so synapses are starting to be lost here. Still, before the earliest clinical diagnosis takes place. And the loss of these synapses is going to have an effect on the networks in the brain so that the functional connectivity will start to change. Again, the earliest change is happening before any clinical diagnosis. And finally, all of those changes will lead now to behavioral changes where the patient or the individual might have learning or memory deficits or executive function deficits. And that's what's allowing the clinicians to make these diagnoses. But by the time the clinicians make these diagnoses, a tremendous amount of brain pathology has taken place. And so wouldn't it be great if we could go back here and find the earliest biomarkers in the cerebrospinal fluid or the blood so that we could alter the trajectory of these disease processes? That's really the goal of Andre and all the neuroscientists working on neurodegenerative diseases. So these biomarkers we can get from the cerebrospinal fluid are specific, they're sensitive, they're measurable, and we can see how those molecules will change to therapy. A huge problem is in Andre, we were unable to acquire cerebrospinal fluid from our patient volunteers. This is an invasive procedure. It requires a spinal tap that can make the patient quite uncomfortable. And all of these biomarkers are in the cerebrospinal fluid, which circulates around the spinal cord and around the brain. And that's a unique fluid area in the body. And the reason is because we have a blood-brain barrier. And that's represented in this figure. So the top part here would represent a blood vessel coming from the heart and lungs. So it's bringing all the nutrients to the brain, oxygen, sugar, assorted other molecules that need to get into the brain down here where the cerebrospinal fluid is. But there's a barrier of cells here, a natural barrier called the blood-brain barrier, and it's not going to let anything in the blood come across into the brain. The brain needs to be well regulated. So only certain molecules can get across this barrier. And the result is that nutrients can flow one way across this barrier from the blood into the brain to nourish the brain cells, and waste material from the brain flows the other way out to the blood to be taken away. Now, these little red circles I put in down here are to represent one of these molecules resulting from one of these proteinopathies. Let's say this is elevated levels of tau in the cerebrospinal fluid or neurofilament light. And in Andri, we were unable to get a measure of cerebrospinal fluid. But some of these small molecules can get across this barrier and are therefore present also in the blood, unfortunately at much lower concentrations, meaning it's much harder to find them. So when we started Andri in 2013, there was no technology to really get after these biomarkers in the blood. But in the last 10 years, there's been a revolution in the technologies that are present that allow us to look for tiny amounts of these small molecules in the blood. And one of these technologies is SIMOA, single molecule array. And this just represents from 2001, when this technique was first discovered, to now the explosion in research publications trying to develop this technology to look for biomarkers in the blood for neurodegenerative diseases. And now it's a simple machine. We have an array here that gets put in the machine. It's ultra sensitive, and it will read the blood of patients and tell us if these tiny molecules are in the blood. And so remember, I told you we had a blood sample taken to do the genetics in Andri way back at the beginning. Now all of this technology has emerged, and we have the opportunity now to do a molecular assessment of what's in the blood of the Andri participants. And we're looking for blood-based biomarkers here. So we collected the blood from the Andri participants in Ontario. Rather than try to create the new technology here, we took advantage of a collaborator of ours, Professor Henrik Zetterberg in Sweden, who's one of the world leaders at trying to find these blood-based biomarkers for neurodegenerative disease. He now has plasma samples in his lab from the initial Andri study, and he's looking for a number of molecular biomarkers in the blood of those Andri participants, including some of them that I talked to you about today. Now let me just switch gears in the last 2 minutes here, and I'll show you the kind of biomarkers I study. So those proteinopathies evolved in the brain, neurons, brain cells got sick, brain cells started to die. Brain cells normally make up these vast connections of networks all over the brain, and these networks are critical for controlling our behavior, our thoughts, our cognitive functions. But when the brain cells get injured and die, these circuits are going to be affected, and they're going to become dysfunctional, and it's going to lead to altered behavior. And that's what happens at the earliest stages in Alzheimer's disease when people start to have memory deficits. It's because the memory circuits here are becoming affected. I happen to study eye movements as part of Andri. This just circumnavigation of the brain shows all the brain areas involved in controlling eye movements. This slide shows the insane set of circuits of all the brain areas connected to control eye movements and blinks and pupil responses. So I'm an expert in this network. And I just want to show you one biomarker that we have from the Andri participants by measuring the size of their pupil while they were watching TV. and we were changing the channels. So this is to represent time. And every one of these little blocks is when we showed the patient a different channel. And at this red line, we went from where the screen was relatively dark to where it was much lighter. So the extreme of this is when you're in a dark room and you walk outside in the sun, it's really bright. One of the first things that has to happen is your pupil constricts. And you compare that when you're in bright light and then go into darkness. One of the first things that has to happen is your pupil has to dilate. And so we set up these changes in luminance in our eye tracking experiment. And I just want to show you one example here where I'm plotting in time the change in pupil size. And this is from a group of 100 control subjects showing when they went through one of these transitions from dim to bright, the pupil constricted. And when they went through one of these transitions that went from bright to dim, the pupil dilated. And when we showed this same experiment to patients with Alzheimer's disease, yes, the pupil constricted on the bright clips and it dilated on the dim clips, but there was a dramatic difference in the pupil response compared to the control subjects. The green line here shows the responses we got from Parkinson's patients. They were more similar to the elderly control participants, but again, dramatically different from Alzheimer's, reflecting the difference in the proteinopathy in these two different diseases. And so I won't go on any further showing you any more of this pupil disease, but just showing you how we're measuring the pupils of the patients and how they're dramatically different. And that's being driven by these proteins attacking these circuits in the brain that control these behaviors. And so I think I'll stop at this time and take any questions if there are any. Okay, so it's my pleasure then to introduce the next speaker, Dr. Bill McElroy, who's the chair and professor in the Department of Kinesiology at the University of Waterloo. He obtained his PhD in neuroscience from the University of Guelph, and he focuses research on understanding how the brain and other parts of the nervous system control movements of the body and translating this knowledge to help individuals improve their recovery after neurological injury, such as stroke or in neurodegenerative disease. And Dr. McElroy co-leads the Andre's Gait and Balance platform, as well as AIM2 and the remote monitoring platform. And I guess the final link I would make from my talk, where I showed you these molecular biomarkers attacking neural circuits that ultimately affect behavior, Dr. McElroy is going to expand on that and show how these different circuits are affecting behaviors that he can now measure with wearables, et cetera. So thanks for joining us, Dr. McElroy.
Thank you very much, Doug. Appreciate the introduction. I'm going to share my screen and I'll just need some sort of confirmation that you can see everything okay. Thank you. Well, thanks for the opportunity to speak this morning. I hope, I know I've been given 2 sections here, and I'm doing this on behalf of a number of other people, and I'll try to break this in the right spot. But Ruth, if you think I should pause for a break, you can just tell me and I'll pause and then we can pick up. I've got a break in here, but it might be too late. So I first wanted to start by acknowledging that what I'm about to tell you about is really the product of a lot of people. And some of the key people to the content I'm going to present are Dr. Karen Van Utegem, Angela Roberts, and Kit Beyer, as well as doctoral students Beth Godkin, Ben Cornish, and some of our key staff, Kyle Weber and Vanessa Tai. All of the folks here, along with the Hands Ontario team and steering committee and all our cooperative students, have been the people that have really made this all work, and all the slides are largely the product of them. And I just have the privilege of being able to present all of the good work that they've done. I wanted to just give you a bit of a framework for what we're going to sort of cover, to give you a little bit of background, why we're trying to measure things basically at home remotely, what and how we should do this, at least from our perspective. What do you have to do to get it right? what we learned today, a little bit about what's in Hands Ontario, and I know Dr. Schwartz will be, talks about this a bit more, but I'll talk a little bit about the feedback forums related to Hands Ontario, and then a little bit about what is coming. So let's just start with the background. Not so long ago, Andre directed some of its attention to this sort of at-home approach, the idea of acquiring and collecting data at home. And the idea was to sort of exploit and take advantage of the emerging technologies that made this possible. Over the last many years, the miniaturization, the smaller batteries, the ability to store and stream data have made it increasingly possible to acquire high quality information from people out of the clinic and at home. And to enable this, to make sure that people with neurodegenerative disease and their care providers and clinicians were part of the target for all of this, we decided as part of Andre, important for us to take some leadership in this space. And certainly we're interested in the role that these remote technologies and monitoring could provide in diagnosis, prognosis, care, and outcomes. And to Doug's point in the talk before, the idea of trying to find basically biomarkers captured in this particular way on sensors, wearable sensors, is to complement all the other measures that are being captured to inform us about next best steps. So I give you this little bit of a framework only to identify where we're going to focus our attention. What constitutes Andre at Home is a much bigger enterprise. There are many moving parts to ensuring that we can collect data remotely. I'm going to talk obviously about the wearable sensors, but for example, there's a significant and important role for intermittent assessing, like cognitive testing. And of course, the Doug's example of eye tracking as a potential target for intermittent assessment done at home, and biosamples. All of those are critical to and eventually an ideally successful at-home approach here. In addition, the idea that there needs to be a very user-friendly interface and mechanism by which individuals can share their information out of home, those are also things we target that are important. And of course, all the related parts, the neuroinformatics, the integration of input from clinicians and research, all of that is part of the bigger picture. But I'm going to focus today on this little window on the left on wearable sensors. And in this space, our goals are trying to take an advantage of what's out there already, the existing technologies and the existing analytics, and try to find the best of those or the combination of those, help remove barriers to use to make them far more usable and useful. And part of this is about how we look at this information. The sensors, I'll talk in a little bit in a minute about what the sensor technology really is about, but it's pretty straightforward. The challenge in all this is what you do with that data. How do you turn it into something useful? And so that's all about the data analytics. So we're trying to make the most of this information by how we sort of analyze it. And maybe the most important thing to do is, what do you do with this data once you get it? How do you communicate this information to the people that are going to use it in a way that's meaningful, whether it be the individuals themselves with lived experience, care providers, or clinicians? We have to convert this data into something that can be acted on. Otherwise, it's just a giant data collection exercise, which might not be terribly useful. So I share this with you a little bit, not for you to read all the words in it, but it sort of captures the scope in the wearable space of all the things you have to worry about. So certainly down here at the end, you need to capture the data at home in some way. And this often involves hardware, sensors, and all important matters of usability and feasibility. Can you capture this data successfully in a way that is not intrusive, that is effective, and that is valid? And then you need to convert that data into something meaningful, some outcome measure that is useful, that is reproducible, that has meaning to clinicians and to users. So this is all about the analytics and what you do with that data and how you move it around. But the part that probably gets a lot less attention is then what do you do with that data? Presenting that data back to people so they can use it. This for us is a really key target in our Andre at Home initiative. And then where we are headed in the future, of course, is how to ensure that it's being used, educated, whole issues around policy and practice, implementing technology as usual care. All of this has to be part of the bigger picture as we move forward and seeing wearable technology and at-home technologies more generally be incorporated into the healthcare system. So this is something you'll see in this presentation that represents one of the major distinctions of what's happening in the Andre at Home approach. Critical to that last part, I would say that critical to the presentation and using is you have to understand who's going to use it and what they need. And so a significant part of the activities we've been involved in is engaging and listening. This is so critical to this exercise. So early on when we started this, we talked to the stakeholders about what they need. And there's this one quote that was quite striking and speaks to our motivation in this space. And I'll just read it to you because it still, it just holds up and is really relevant when you're thinking about capturing information at home and why it might be important. Living as a caregiver 24/7, each small change in your loved ones become the new normal. And you can easily miss the more day-to-day changes as you're looking for the big changes that you can report to the neurologist at your scheduled appointment once every 6 months. And this speaks a little bit to the opportunities that are afforded collecting data more continuously, more sensibly to detect changes and track things that is one of the reasons we think an at-home approach might be particularly useful. Of course, people had lots of particular areas they thought would be important and interesting to them. And we certainly tracked those and tried to marry our approach to the needs of the people that we're serving in this space. So why measure at home? As I mentioned, there is a demonstrated link between health and measures we can capture at home. And I'll give you just a couple of examples, and there's many more. One very strong example, and this is work that we get great guidance and leadership from Dr. Andrew Lim, and that is the relationship between sleep and risk of and progression of a neurodegenerative disease characteristics, particularly those related to dementia. Sleep is an interesting and important behavior that is obviously much more difficult to capture clinically. It can be done, but if one is able to do it successfully at home, it certainly makes it far more practical. Mobility is another one that has gotten lots of attention about gait quality and movement emerge as an early preclinical feature of cognitive decline. And this is another area of interest in some of the measures we capture about what mobility looks like and the patterns and the characteristics. Another one that's emerged very strong over the last 10 to 15 years is how critical physical activity is in reducing or influencing risk of dementia. And then one area that Dr. Andrew Roberts helps lead us is in the whole idea of social connectedness and how it's connected to exceptional cognitive aging. In this particular case, the important issues such as speech and language as markers of social connectedness are an area that we have a strong focus on. And the other reason to be at home is some of the measures we're interested in are very difficult to capture in a clinical setting. I mentioned sleep already, cardiovascular function, dysautonomia, activity and mobility, and socialization, and agitation, anxiety, stress. All of these characteristics or features or physiological responses to situations are hard either to simulate in the clinical setting or may not be captured in the short time someone is in the clinic. So being able to capture these things at home is certainly a significant advantage as we sort of uncover the relationship between the disease, the progression, and even links to treatments. So measuring home can provide a few things for us. It can provide a more rapid response to action if we're capturing data more sensibly and more often. It has the potential to reveal causes of things that are infrequent or time varying changes. So things that change over time within a day or they're relatively infrequent, we can capture the data that might reveal the cause of those. There's evidence that is a more sensitive measure to evaluate some treatments because you're evaluating that data on almost a moment-to-moment basis related on the timing, for example, to particular treatment. A good example of this is the work we're doing as part of an impact study in the relationship between Parkinson's medications and characteristics in day-to-day life. And another really important one is the opportunity that wearable devices and remote measurement can help serve those who are underserved by clinical services, those who have reduced access, either because of geography or other challenges. A remote measurement technology has an opportunity to help them. So what and how do we measure things remotely? What are the approaches we can use? Well, the new technology, as I mentioned, has made many things possible. And there are devices for a number of things that can capture physiologic responses, movement. There's all sorts of devices around wearable locations that can capture motion, cardiac function, muscle function, skin conductance. There are other devices around measuring body motion. All sorts of things exist now that allow measurement at home possible. And they're small. They capture the data over extended periods of time. And this has accelerated over the last 5 years. If one tracks the number of papers in the literature related to wearables, it is logarithmic. It is just climbing so quickly. And it makes it a wonderful opportunity, but it also makes it quite complicated about deciphering what is working and what is not. The ones you'll may be most familiar with, or most people familiar with, are the consumer ready ones, the Fitbits and the Apple Watches, and they've had a lot of attention. And a lot of those devices are being used, at least for health applications. They certainly are a convenient wear location, worn on the wrist. They do provide some summary health measures, although at times we worry a little bit after we've sort of validated them about the quality of that signal. And it can tell you a little bit about what you did compared to your health status. So they're interesting tools. What they are is a demonstration of the possibility. That's what I think they really are. They are really quite cool demonstrations of the possibility of sensors worn on the wrist to acquire data continuously. But as a tool to inform us as we focus on neurodegeneration, either risk of or progression, they really are very limited. And I'll share that with you why in a moment. There are a number of what we call research-ready wearable devices, not as appealing in terms of maybe what they look like, but are capturing the data in ways that are important and unique from those commercial devices. In other words, the data is available to us. I'll talk about that in a minute. But they all have the same feature. They all contain the same sort of sensors, which makes them all possible to collect the same type of data. So we're going to talk just about a couple that are common to the devices, all of these devices, and a focus of the devices that we extract behavioral data from. Accelerometers and gyroscopes are what are classically called inertial measurement devices. They're based on inertial properties or motion properties to capture the motion of a device. An accelerometer measures linear motion and linear acceleration specifically, and a gyroscope measures rotational velocity. And so if you place them on the limb, you can measure movement. And if you place them on certain limbs in certain places, you can get quite specific about the nature of that movement. There's also optical sensors. Optical sensors are the ones you will see easily see in a Fitbit, for example, or an Apple Watch with the lights that are on the bottom. Those optical sensors emit light and then have a collection device on the other side to measure the changes in tissue properties or fluid motion. So they'll often be used for pulse pressure in PPG systems or oximetry for oxygen levels. So optical sensors are quite common. Bioelectric sensors, we focus on a bioelectric sensor as one of our sensors to measure cardiac function, ECG. These are just devices that record the difference between electrical potential. So you could have a bioelectric sensor for muscle activity, for galvanic skin response, arousal, that sort of stuff too as well. But what's fundamental about all these is there's just sort of, there's foundational sensor types. And you will find these in almost, in many, many different devices. The watches, for example, the Fitbit watches have accelerometers, gyroscope, optical sensors, thermometers. The Apple Watch, the new one, has a bioelectric sensor. When you touch the crown, it closes the bioelectric loop and you can measure some cardiac function. So all of these you'll find in conventional and research grade tools. And I thought I'd take the liberty just to give you a bit of a sense of what are involved in these. So this is just demonstrates to you, this is a Gene Active Watch, one of the original ones we used. It's a research grade device, has a 3-dimensional accelerometer in it. I'm just going to run a little video. And this video is showing you when you rotate the position, the acceleration changes due to gravity. And you'll see these shift in its position. So these slow frequency are just showing the different tilt positions as gravity pulls on the different 3-dimensional sensor. And if you do it something more dynamically, depending on the direction you do it, you'll see the motion in this device. And that's all an accelerometer does. It conveys the information about the linear acceleration of a device and its orientation. Now, what's really critical is where you place this device. It gives you either a really good ability to detect a specific behavior, or it's more difficult. So this particular video captured on behalf of by Ben Cornish for us just shows you this. He's gone out and captured some data on a walk. And I'm going to pause this just for a second. So he has one on his wrist and he has one on his ankle, and these are synchronized. So they're happening at the very same time. And you'll see here on the ankle, a really clear pattern. No surprise here. That really high impact that you can see here in the red here it is in the blue and red, that really high impact is the foot hitting the ground. When you hit the ground, there's a high degree of acceleration, a shock wave actually that runs up your shank and body. It's a kinetic characteristic of walking and very distinguishable. And as your heel comes off the ground, you go through a swing phase and you can see the linear acceleration of the swing phase. So you see this very reproducible pattern, one limb after another alternating. If you're going to measure walking and characterize the features of walking, either looking for step-to-step variability as a variability index of dynamic stability control, which we know to be an important indicator of central nervous system status and linked to neurodegenerative disease, or if you're interested, for example, in how much people walk or the volume or the characteristics, the ankle is a place to be, or at least on the leg. At the arm, you will see a rhythm. You'll see an oscillation in here that occurs as the arm swings. And if you do some analytics, you can find the cycle of walking in here, but it's not nearly as strong a signal. And it's much more difficult to be reliably detecting. And what if you're holding on to something? Of course, then it becomes not very useful at all. So while an accelerometer can be a very powerful tool, where you wear it and how you analyze it ultimately define its usability. And this one, Beth Godkin grabbed us a bit of data for us. This just emphasizes the point even more. This is just her stationary cycling, and it just emphasizes the importance of wear location. Of course, there's no surprise the arms are not moving here, but she's very active and you can see the rhythm. And you'll also notice the pattern is different here. We can distinguish, obviously, cycling from walking here because the pattern is so very different. So how you use these devices and where they're worn is pretty critical. And I'm going to walk through here and just show you a couple of examples of how you could just take the principle of accelerometry and reveal behavior. And this is all based on where you put the device and how you analyze it. It's the same sensor, the very same sensor is being used here to extract very different things. So as I said before, mobility has, walking has this very stereotypical kinematic and kinetic characteristic. And you can see this pattern on a leg worn very simply. It's the same sort of idea I showed you on the last slide, where you see this very consistent and regular pattern, a stance phase, a swing phase, and impact. And this continues and iterates as you walk. And this data here is, this is a little bit hard to follow, and I just meant to be something we go through quite quickly, but this is 7 days worth of data from an accelerometer. This happens to be a gyroscope, but the same related principle, I suppose. But this is 7 days worth of data. I highlight this to you because I want to zoom in. Right here, you can't see much. You can't distinguish much, but what you can see is circadian. There's a sleep, there's a wake, there's a sleep period. You can see that pattern, but you can't see much else. If I zoom in a little bit, I've zoomed in here to probably about 12 hours or more. Here more easily, you can see the sleep period here, and then they've gotten out of bed here, They start to move a little bit. Again, you can see the patterns at a macro level. It gets increasingly interesting about what you can pull out. If I zoom in a little bit more, you start to see bouts of activity that are a little bit more specific, sort of high intensity bouts here, and there's one here I'm going to zoom into in a minute as you start to zoom into this data a little bit more. And then I zoomed into this period even more, and I've done this for a very particular reason. If I go back, this is the period I'm looking at, this bout right here in time. It's not a very long bout, but it's very specifically walking. This is a bout of walking. You can see the gait initiation starts right here. There's a gait termination phase. This is a relatively long bout in terms of number of steps, but what's underneath this is a very specific pattern. Identifiable and distinguishable, extract the gait information. We know it's walking. I know that's walking because of the wear location and also the signal characteristics, how we analyze the data. We can also use accelerometry to estimate the intensity of activity. Kyle Weber's been doing a lot of this work for us, looking at how you use accelerometry off the leg of the arm to characterize how intense the activity is. So I can then take, here's some data on this side here, again, 7 days worth of data. You can see the circadian cycle, wake, sleep cycle, all the way through this for the 5 to 7 days. And then what's interesting is the intensity of this activity is a measure of how active somebody is. And that's a body of work that's been going on for us for a bit of time, because we know the relationship between physical activity and neurodegeneration. And there's a number of approaches you can use to estimate activity, again, from the same sensor. You can use the same sensor, accelerometry, thank you, Dr. Andrew Lim, for these slides, in the same way to estimate sleep quality. And we do this in our studies to use accelerometry data, estimate things like sleep duration, sleep fragmentation, as well as I mentioned before, the diurnal rhythmicity or characteristics of sleep. And you can do other measures to augment sleep, but accelerometry, Warren, in this particular case, most commonly used on the wrist to estimate activity, is considered a very important and reliable index of sleep quality. You can interestingly also use accelerometry to measure speech, and Dr. Angela Roberts, who's a key part of our Andre team, leads this very innovative body of work with her colleagues at Northwestern University. And in this particular case, the sensor here shown on the left as an example of this is worn just around the sternal notch, just below the vocal cords. And when you speak, those vocal cords vibrate. And when they vibrate, they cause motion of that sensor. Remember, that sensor in there is an accelerometer. It detects any kind of motion. It's just now really, really small motion. And you can see vibrations and mechanical acoustic vibrations. So accelerometry can be used to measure speech. And Dr. Roberts has a remarkable toolkit that helps extract out features of that talking with regard to the complexity of speaking, that gets us back to social connectedness, cognitive biomarkers. You can also extend this accelerometry then to swallowing. And some ventilation information as well in terms of breathing. And these are all important things as we look into other characters such in ALS, where swallowing, for example, is a very important marker of the progression of the disease. So accelerometry, again, properly worn, analyzed, can yield a lot of meaningful information. And more recently, we've been looking at using this to the same devices, using it to assess posture, lying, sitting, standing. And the reason for doing this is the important link between sedentary behavior, trying to minimize sedentary behavior and the impact it has on brain health. And so you can use these devices, particularly if you combine them. A trunk and a shin, for example, combined together gives us a high probability of predicting. Kyle Weber's been working on these analytics for us, but this is a really good example both of the value of accelerometry and merging sensors, or something called sensor fusion. Mentioned this one. Now we drift a little bit from accelerometry, where I hope you get a sense that there's a lot that's possible about markers. And I haven't mentioned the other utility of the IMUs. I was going to do that at the end, but I should make sure that it's clear that we don't just use them for walking, we use them for tremor detection and bradykinesia. We look for other characteristics or symptoms that are movement related. This is what the IMUs can afford us. The bioelectric devices we've been using have been principally around measuring cardiac function. And of course, ECG is the electrical activity of the heart. In this particular case, it's really just a sensor that's chest worn, either vertical or horizontal, and you can generate this very classic ECG profile. And an example of some of this data that we've been using is the raw ECG signal, and you can extract heart rate, for example, from these. The red dots automatically detected the heart rate from the ventricular contraction that's occurring. And then you can map that heart rate over a 24-hour period. So here's the changes in heart rate over 24 hours. And you can look at it and look how variable the heart rate is. You can look for changes in heart rate that are meaningful. But the interesting part of this is if you don't have context about what's going on, it's somewhat difficult to interpret the changes in these heart rates. So context is absolutely critical. And how do you get context? You get context by combining other sensors and other behavioral markers. So at the very end, here is an example, and I'll show you another slide in a minute. It's just a bout of walking, where you can see the elevation in the heart rate here that's occurred at this period here was temporally coincident with the period of walking. So when they began to walk, their heart rate elevated. And this is really important for us as we start to look for how heart rate and gives us an index of things that are happening. So anxiety or stress that is an increase in heart rate that would be dissociated from a movement requirement is something we will look for. An example here of the context is the gray represents on the graph. This is a much shorter window. It looks like it's about an hour's worth of data. Kyle passed me these slides the other day. So as he's processing some of this data, so there is heart rate over time. This is just the heart rate changing over time. And all the gray bars are periods of walking that are overlaid on top of it. And you can see the relationship at times of the clear relationship in this bout, for example, where the heart rate response immediately responds in a very workload dependent and then drops at the end of the workload. You can see other times where that relationship is not as clear, but there's this important coupling between heart rate, movement, and then non-movement. This is a good example of one on the bottom to say, what is the context here? The bottom graph here is a heart rate that's continuous collected over a much shorter period. We've got a few minutes worth of data here. And then there's this large steep rise in the heart rate right there. And one could be somewhat stumped about this is if you don't have other information. Well, this is actually pretty simple. This is someone waking up in the morning and getting out of bed. And the reason this is such an important example to show you is we're particularly interested in dysautonomia, the idea of regulating blood flow, particularly as it potentially relates to falls, but certain autonomic function in its relationship between movement and the regulation of blood pressure is sort of critical. And we can see these trends in. So if we overlaid posture here, you would see the postural transition and the heart rate response during that. So this is just to reinforce the idea that having a sensor is one thing and having lots of data, but without context and without interpretability, you can be left just with lots of interesting data.
And then another question that just came up is, can you briefly speak to the bioethical implications of at-home monitoring?
No one can speak briefly on this topic. That would be extremely difficult because it is complicated. But I can tell you that remains one of our most important sort of guiding principles. And one of the things we've been always advocating for is this idea that what we're really doing is trying to collect data that empowers the users. So the data is, theirs to use, and then we share it appropriately with relevant clinicians. The monitoring of people continuously is always a challenge. Certainly all the issues of privacy are a major. I did want to speak just quickly to the whole issue of the attention that's paid to this issue around ethics and all the issues around of that. A good example of one that came up, and this is probably more about privacy, is this whole issue of speech detection and the idea of audio recording and all the implications that would lead to. And of course, Ange Roberts, involved with the group on speech, this is sort of gets resolved in this very unique way she's approached it, in which it's a mechanoacoustic sensor. In other words, no sound is collected at all. And so at the forefront of all of our concerns, always as we proceed through this, are the ethics, are the privacy of the data, of where it goes and who holds it. And I haven't done justice to that excellent question, but I can certainly offline chat more. Thanks. So I in the limited time that I have, I'm going to expedite, I'll flip over some slides, and I wanted to address a few things. First of all, the approach that we're adopting in Andri is to try to get this right. And there's a few principles that we've sort of focused on. First of all is the importance of the data to the needs of the users and the care providers. This for us is absolutely critical. If the information is not useful to the users, care providers, and clinicians, then it's not really going to be helpful to make a difference. And the other thing is it really has to be usable, personalized, and of course it has to be validated. All of these are absolutely critical, and the usable has to be really usable. It's one day say someone can use it. It's another thing to say they can use it for 7 days, 10 days, not encumbering them. And there's really two different levels of usability. We've really focused our attention on making things innocuous, not difficult to use, and therefore we've got devices, for example, which people just basically put on and ignore as to an attempt to achieve that. And that's something that's absolutely essential to the success of this, and it does relate to one of the observations we've noted already about the effectiveness of this. As I mentioned, we've identified interest in stakeholders, and so it always came back when we started, why not just use a Fitbit or an Apple Watch? Well, the measures that we're interested in are not available off of a Fitbit or an Apple Watch. The measures that are related to particular diseases or symptoms, you're just not going to get them off an Apple or Fitbit. You can't tailor the experience for the user. And bigger concern is you can't advance the science because you often can't get the data. Now, Apple does have an opportunity for people to collaborate with them, and you can, but often the data is hidden and often the data is proprietary. And the other thing is, putting a device just on the wrist, I hope gathering from what I said before, is pretty limiting. The activities and behaviors we can capture off the wrist is not actually an ideal location for many of the things we want to capture. So the single sensor off the wrist, not ideal. And actually more generally, a single sensor is not ideal. And so then the question becomes is what sensors and where do you use them? So we have a multi-sensor approach, which is somewhat unique to our work. This multi-sensor approach, we still look to minimize the number, but maximize the benefit. For example, in the Hands Ontario, we have 3, one on the ankle, one on the wrist, and one on the chest, providing complementary information and provide us the various measures that we need. Without those 3, it would be very difficult to reconstruct meaningful behavioral measures that would inform us. This is what's possible when you have multiple sensors. You can get all sorts of measures from single sensors, and then you can combine these sensors for other things. And we really view this as an essential step forward in the use of wearable technology. It just can't be a wristwatch. What have we learned then? Because I've just sort of quickly through there made the point that it really have to have a multi-sensor approach, and you have to tailor it and personalize it as we learn more and more. But we have been doing this for a little bit now. We've had 3 studies, an early pilot study in individuals who are average age was 90. They wore 4 sensors 24/7, multimodal and multinodal. Then we had our feasibility study in people with lived experience with neurodegenerative disease, wearing as many as 6 sensors for 7 days. And then we've been doing a feasibility and usability in a PD and dance study where we're looking to track not only daily life, but also the intervention. And all of these studies, a key component was feasibility and usability. Were people willing to do this? And part of it was about how we could develop an approach that would permit it without people getting frustrated in too many devices. We've landed on Hands Ontario on 3 because we've been able to minimize some of the need for all of those sensors, because in these early days, we're trying to learn lessons. One of the important messages here of these various sensors we use, ankle, wrist, and chest are the ones, is that people are willing and able to do this. These are our non-wear periods. We use non-wear as periods when people took it off as an indicator of tolerance and feasibility. And a recent paper that Beth Godkin has led with Karen Van Utken is about the feasibility of multiple sensor wear in people with neurodegenerative disease. And it was highly accepted. Down here, you can see a couple of people down here who have higher non-wear periods, but the majority of people are really just taking them off for a shower or a bath. And for 7 days, 24 hours a day, willing, and at the end of the study saying, yeah, it was not a problem. And part of the reason is because they don't have to do anything with them. They put them on and take it off. The key factors, however, were comfort, ease of use, can't interfere with my activities of daily living, and an important matter of aesthetic, which we still haven't mastered quite as well. But these are all key criteria to ensuring people are willing and able to use them. Now you have to remember, they're not interacting with these devices in a meaningful way. They're really placing them on, and all the back end is getting done without them having to worry about it. The other part that we learned about and we developed is feedback. This is back to the idea of getting information. So in the Remind study, we developed our first feedback form, the form that goes back to users to give them information. And we did a lot of work on the front end about what this information was and how we should do it out of the Remind study. And it has led us to develop in the Hands Ontario a very elegant starting point for our V1 version of the feedback form, which I'll show you in a minute. The other thing that we've been able to do is reinforce the idea of personalization. This is 6 randomly selected people out of Hands Ontario, literally yesterday, randomly selected without regard to who they were. And this is, it's a bit hard to see here, but these are different days for each person. So this is 2, 4, 6, 7 days. And the pattern up here is the daily pattern of walking, 7 days for one person, then a second person, a third, fourth, and fifth. And the thing that will strike you is the variability between people and between days. One cannot imagine a single solution and an absence of personalization to be a win here. We have to be able to personalize. We have to be able to give back information that is unique to them. And we have to look at solutions that address the heterogeneity of behaviors and characteristics. So I'm not going into the deep dive into Hands Ontario, but I'm going to highlight a couple of things. I want to highlight the Hands iceberg, I have now been calling it. And the iceberg, of course, you see lots above the water. You don't see anything that's in behind it. And so what is collected in Hands Ontario is the things I mentioned before: an ankle worn IMU, that's the inertial sensors, or wrist worn, a chest for an IMU and ECG. Those are, they wear 7 days, innocuous. We're eventually incorporating speech. That's not done in our currently Hands Ontario. It's been done in some of our impact studies, but those are all collected sort of continuously. Blood samples, clinical work, health comes and service. See all that on the end. And what happens at the beginning is we do an analytic pipeline that gets us to gait, sleep, mobility, and sedentary behavior. And we generate this wonderful feedback form. And it gets a lot of attention because I think it's extremely valuable as a starting point. The thing that someone has to always understand in all of this is that is the tip of the iceberg of what Hands gets us and what Hands has the opportunity to do. That's my attempt at a boat, by the way, that thing in the middle there. I know it's a little weak, but that was my drawing attempt with my mouse. So what's under all of this iceberg is so much more that we're continuing to advance what the Hands Ontario experience is doing. So all of these other measures, either in development or part of the clinical report, which is under development, cardiovascular, anxiety arousal, speech, falls, posture, so on and so forth, biomarkers, genetics, cognition, clinical outcomes. What's underneath this, of all of the stuff about advancing the clinical report and advancing knowledge, all the things that Doug and Mary were talking about is all part of Hands Ontario as well. So the wearable sensors and the feedback form get a lot of attention and they're visible and very important. But I think it's critical to understand what's really under this is all of the bottom of that iceberg. I'm going to skip that. I just want to talk briefly about this feedback form because it is one of the key things that we've been developing as a way to share the data. The feedback report that's provided as part of Hands Ontario is an augmented one developed out of the Remind study. It has some very nice information for people just as a resource in terms of readability. It's been laid out in a way that provides both tabular data, which is high-level tabular data information with regard to guidelines that are in the literature. It does daily summaries. It provides day-to-day variation in terms of patterns. The modularity of it and the readability of it have been augmented through feedback with our PCAC and others to try to get this right so it is usable. Some sample images from an example of the feedback, which is delivered both electronically and in paper as required, is looks something like this for walking. So we do get that step count thing. Your Fitbit would do that too. But again, it's not all we get from this, and it's not all we display. That daily summary that's out here, which has your step counts, has the daily patterns, and then long bouts and extra long bouts, because a lot of the steps will be activities of daily living. Our interest is in augmenting that with some purposeful walking to improve activity. And then you can see the patterns, how they were distributed over the day. This is important to us because we're looking for relationships in day-to-day variation and when people do and do not engage in activities. And then once we've got this, what's the characteristic of that walking within that day? It's something we can do with the sensors that we have. Same thing for sleep, looking for sleep fragmentation in these patterns. When you get up at night, all of those things are possible, and we deliver back on these feedback forms. And daily activity patterns and sedentary behavior in the same way. These are all serving as our first formal return for information that's done as part of the HANDS Ontario study, which is followed up, interestingly enough, by what we call our V5 visit. The V5 visit is our attempt to get feedback on the feedback so we can continue to improve that part of what we're working on. And so 2 weeks after receiving the feedback, individuals have a V5, and Dr. Angela Roberts and Karen Van Uttingen put together these slides for me, which is just a quick highlight of some recent, the first 14 people, I believe, that we've had a chance to do this. So 92% of the people found this information to be beneficial to varying degrees, 52% very helpful, 21% helpful, and 50% somewhat helpful. But 92% are finding some value in this. And I think I spoke to Karen this morning about this, and there really is an appetite to get information back in a timely way. So these are coming back to people in a 2 to 3 week window. They're not waiting for 6 months when the study is done. These are done because we've automated the pipelines, and they're coming back quickly to people to talk about what their data looks like. 38% of the people made a change to lifestyle within the last 2 weeks, in part because of the information they gathered from this. They were increasingly aware of good sleep and monitoring sedentary behavior. 92% shared the information with a family or friend. Critical, I think, in this whole exercise of it, sharing health information with others, having the conversation and talking. The motivation to share was related to making lifestyle changes and educating, advocate change in others. They were also using their information to encourage others to attend to these important matters too as well. So a variety of reasons. Saw your hand, Ruth. Thanks. Almost there. And then, of course, the motivation for sharing with family and friends is around self-advocacy, trying to help partners understand their symptoms, helping other family members understand their symptoms. And again, remembering that the clinical reporting is all part of what's to come. This is just a sort of the start of where we've begun. So I wanted to get just really briefly in 1 or 2 slides, it just takes just a couple seconds here to wrap up what's on the horizon for us. Certainly the clinical report is a real target for us, is now getting those disease specific characteristics as part of our standard reporting and refining those analyses, which are ongoing right now for tremor and restless leg syndrome, freezing of gait and bradykinesia, dysphagia, dysautonomia, agitation, and cardiovascular events. All of those are the target of our analytics. Refining those that exist in the literature or developing our own. And we still remain importantly focused on technology that's emerging, because we're not doing this in a bubble. People are developing technologies, and we're looking for ways to integrate these and at-home assessments that allow us to do intermittent testing, such as the kinds of things Doug mentioned, and cognitive testing done in other ways. All of that is part of this bigger package and the target of what we're headed to. Hope to much more from a small number of small sensors to achieve a lot, maximize the benefit of very little challenge and effort on the user side. Anyway, I'll pause there, Ruth. Sorry, I went a little bit fast at the end there, but hopefully that'll give you a flavor of what we're doing.
That's perfect. Thanks so much, Bill. Really a great presentation. Great, and Yuan shared. Thank you very much. So it's my honor to introduce us to the next presenters. Susan Bronskill is a senior scientist and scientific lead of the Life Stages Research Program at ICES and a professor in the Institute of Health Policy, Management and Evaluation of the Dalla Lana School of Public Health at the University of Toronto. In her research, Dr. Bronskill makes use of population-based administrative databases to study transitions between healthcare sectors and focuses on improving quality of care, medications use, health services utilization, and healthcare outcomes for older adults, particularly persons with Alzheimer's and related dementias, women, and those who are frail. Dr. Bronskill leads Andre's AIM4. And then Laura McLagan is a senior epidemiologist at ICES, where she designs, analyzes, and reports on health services research studies using large administrative databases. Laura is the portfolio manager for Andre's AIM4, our health services area that will be discussed right now. In this role, she manages a series of health services research studies focusing on health services use and costs, prescription medication use, and outcomes for people living with dementia, Parkinson's disease, ALS, and stroke in Ontario. Laura holds her MSc in epidemiology from McGill University and a BSCH in biology from Queen's University. So welcome.
Thank you, Ruth. We're delighted to talk to everybody today. And what we're going to end up doing is a lot of what we've talked about so far today is focused on research on individuals. So taking individual measurements from people, whether they be clinical or sensors or things like that. What we're going to do is take the higher view, and we're going to think about how we characterize all 200,000 persons living with dementia in Ontario. So next slide, please, Laura. And the technique or the research tools that we use to do this are a type of data called health administrative data. And so what we're going to do in our presentation is talk to you all a little bit about what these data are and why they're useful for research. I'll do that part. And then Laura is going to share some research findings from 3 studies that we've recently completed, so you can get a sense of how the data are used in action. And then we'll have a few takeaway messages. We'll tie this into how the health administrative data are going to be used in the HANDS Ontario study that you've heard mentioned a lot today. And we actually have a few questions for you, although we're tight on time, so maybe we'll wait until all the presenters have gone. So when we're talking about health administrative data, we're talking about data that are generated by governments and other organizations for healthcare billing, record keeping, and evaluation. So you can think of this as in Ontario, each time you go to see a physician, you visit the emergency department, you have a drug prescription filled, that can be considered a health system encounter, and there's a record of that health system encounter. And those healthcare system encounters exist for nearly all 14 million Ontarians. And so what we do as researchers is we're able to use these data and link them across people in a de-identified privacy protected setting. And that lets us conduct studies on populations and how populations use health services. Next slide. And we do this at an organization called ICES. And this is a little video that's just going to describe health administrative data and for you.
Since 1992, ICES has been working behind the scenes to improve Ontario's healthcare system and the health of the people of Ontario. How do we do all this? Let's break it down a bit. The Ontario government collects routine information about healthcare system use so it can plan, administer, and pay for health services. All this information is called administrative data. A new piece of administrative data is created every time someone has an interaction with the healthcare system. Whether it's a visit to the doctor, a prescription being filled, or a hospital admission, every piece of information is important. Usually, this data is stored in separate databases, but the real power of administrative data comes when we link them together to create a big picture of healthcare patterns and trends over time and across Ontario. Linking the data lets us look deeper, way beyond the individual picture. It allows us to identify problems and evaluate potential solutions that will improve healthcare for the whole population. We go to great lengths to protect this important data and the public's privacy. In fact, the first thing we do is to remove names from the data before it is used for research. Through our innovative use of administrative data, ICES generates powerful evidence that is used in the planning and evaluation of Ontario's health system, evidence that supports better policy, a stronger healthcare system, and healthier people.
So I find that that video manages to present the concept of health administrative data much more eloquently than I can do on my own. So hopefully that was helpful to people. We talk about ICES, the video talks about ICES, so that's an independent, not-for-profit research institute where our core funding comes from the Ontario Ministries of Health and Ministry of Long-Term Care. Many other organizations have access to the data as well, sometimes in different forms. So the Ministry of Health itself uses the data for analyses. Many of you might have heard of the organization CIHI, which collects a lot of the emergency department and hospitalization data initially. So we're just one player in the system of who uses data. We're a community of research and data, as well as clinical experts. And we have a secure array of Ontario's health-related data. So the core of our data are the administrative data, but there are a lot of studies that happen that use electronic medical record data, population-based health social survey data, clinical cohorts get brought in and linked to the data. So the HANDS Ontario study would be an example of that. And we're an Ontario-wide network. So the picture you see here is our building on the grounds of Sunnybrook Hospital. Those of you who live in Toronto may have driven down the hill behind Sunnybrook to go and walk your dogs at the dog park that's down there. That's us on your left as you're driving down the hill. But we also have sites at all of Ontario's medical schools, and they're listed there. Next slide, please, Laura. Oh, back one, yeah. So I think you saw Mario present this schematic earlier in terms of the different aims of ONDRI. And so we connect in with aim 4, which is here in the bottom left. And we're really focused on analyzing the health system records to inform healthcare policy and planning. In consultation with members of ONDRI's patient and health partner community, we identified some key themes where we wanted to anchor our ONDRI aim 4 research. And they're listed here. So we have conducted a number of studies looking at health system use and cost of services across settings. We have a theme that focuses on multimorbidity. So obviously individual conditions like dementia don't occur by themselves. They frequently occur with diabetes, with Parkinson's disease, with other sorts of conditions. And so we have a series of studies that looks at characterizing this multimorbidity, but also thinking about what kind of health system multidisciplinary approaches are needed to address them. We have a series of studies looking at medication use. Obviously, there are some dementia-specific medications, but also medications that are used in older adults more broadly. And then we're also very interested in trying to think about how to extend the administrative data with rich clinical cohorts, with electronic medical record information and other kinds of studies. So the examples that Laura is going to talk about today fit into each of these themes. So she's going to present you some findings from one of our recent studies looking at the impact of COVID-19 and health service use in persons with dementia and Parkinson's disease. She's going to share some findings where we look at what other conditions co-occur and the sequence of that co-occurrence with dementia. And she's also going to share some findings on sex differences in how antipsychotics and benzodiazepines are used in the long-term care environment. And finally, just briefly, she's going to touch on how we connect the health administrative data into the HANDS Ontario research study. So over to you, Laura.
Thanks so much, Susan. Hopefully you can hear me.
You're a little bit quiet.
Oh, I'm a little bit quiet. Okay. Okay, so I'll try to speak up. So it's a pleasure to speak to you all today. And as Susan mentioned, we're going to be providing 3 research snapshots to give you an idea of some of the work that we've been doing in Andre AIM 4. And the first one is relating to COVID-19 and healthcare services for people with dementia in Ontario. So we all know from our daily lives that the COVID-19 pandemic has really disrupted the way we access healthcare services in our province, across the country, and globally. Now, the pandemic we know has placed an enormous strain on the healthcare system, and it's also changed the way that people with dementia and other health conditions interact with the healthcare system. And we know from previous work that people with dementia can be more vulnerable to disruptions in important healthcare services like home care services. So in this first study, which is shown on the right-hand side here, we investigated how the pandemic impacted the delivery of publicly funded at-home home care services, including support for personal support, nursing services, and services for therapies like physiotherapy and occupational therapy. So overall, we found that home care visits for personal care during the pandemic for people with dementia declined by 16% at the lowest point during the pandemic, but actually rebounded during the fall. So you can see that by the shape of the graph here that we're showing. In the 3 graphs, we've sort of separated these trends by different groups of people with dementia. So we've shown people living in rural and urban areas on the left-hand side, people of different sex, so males and females in the middle, and people residing in different levels of income neighborhoods shown on the right-hand side. So what we found was that people living in rural areas generally exhibited a slightly larger decline in home care services for personal care during the pandemic than people who were living in urban areas. We found that men and women had similar declines in home care service use during the pandemic, and those living in the highest income neighborhoods actually had a larger initial decline. The good news is that the home care services relatively quickly rebounded to what they were historically. So the period of a low level of home care support was relatively short during the pandemic. But of course, this still raises important concerns for people living with dementia and their care partners. So next, during the COVID-19 pandemic, we also wanted to examine how people with dementia interacted with other healthcare services. So things like emergency medical services, so ER visits, as well as hospitalizations. And we know that hospitals really play an important role in providing care for people with dementia, so this was important to look at. So we asked the question, how big of an impact did the pandemic have on the use of these healthcare services? And we compared patterns in 3 different patient groups. So we compared them in people with dementia, people with Parkinson's disease, and older adults who were age 65 and older who weren't living with these conditions. And what did we find? Well, what we found was overall there were declines in many of the healthcare services that we examined. So here you can see the rates of emergency department visits for people with dementia on the left-hand side, Parkinson's disease in the middle, and older adults on the right-hand side. So we found that overall there was a substantial decline in the rate of emergency department visits, ranging from 55 to 60%, depending on the group we looked at. And this raises important concerns in terms of care that may not have been received, urgent care that may not have been received because of fears of presenting to the emergency department due to infection and other issues. The rates did rebound, however, by the end of this study period, which was towards the end of September. So next we looked at family physician visits during the COVID-19 pandemic, and I'll walk you through how to interpret this graph here. If we look at the graph here, we can see the total number of visits during the 2020 year, which is shown by this dark line, solid line here, and a dashed line that's in lighter blue indicates the rate in 2019 for a comparison. So overall, this white space here, you can see in the dementia graph, in the Parkinson's graph, and the older adult graph, indicates that there was a decline in the rate of family physician visits overall. But what we found was there was an interesting pattern when we looked at virtual visits versus in-person visits. So we can see the virtual visits are shown by the darker gray here, and the lighter gray indicates in-person visits. So at the start of the COVID-19 pandemic, which is this orange line here, we saw that across all groups, there was actually a very rapid shift to virtual care. And this is showing virtual family physician care. In-person care, of course, dropped, but predominantly virtual care sort of took its place, and some of the rates even increased to higher than historical levels by the end of the study period. So what does this indicate? So this indicates that this is a potential success of our health system in adapting to the COVID-19 pandemic, in that we were able to maintain this vital family physician support using a different means, using virtual care. And we'll come back to that a little bit at the end as we have some questions for you if there's time. All right, so that was our first snapshot, really focusing on COVID-19. And as Susan alluded to, there's also some important work that we've undertaken looking at how dementia develops along with other conditions that people with dementia might have. So we know that people with dementia often don't just have one chronic condition. So they may have dementia, as Susan mentioned, they may have hypertension or diabetes or Parkinson's disease. And in this particular study, we wanted to understand, how do these diseases develop? Do they develop before or after the dementia diagnosis? And how does the development of dementia influence the risk of subsequent diseases? So in this study, we looked at over 5 million Ontarians who were aged 40 to 85 years in 2002, and we followed them for up to 14 years to understand this pattern of neurological and psychiatric conditions. So in this snapshot, we looked at the relationship between the timing of a dementia diagnosis, which is shown on the left-hand side here, and the association with a mood or anxiety disorder after a dementia diagnosis. So what did we find? Well, overall, we can tell from the position of these dots, which indicate an increased rate of mood and anxiety disorder, the further right they are, we saw that within the first 6 months of someone having a dementia diagnosis, their rate of developing a mood or anxiety disorder was very, very high compared to people who did not have a history of dementia. And we saw that this relationship was actually sustained across different time periods since the dementia diagnosis. So what does this mean? So this indicates that mood and anxiety disorder is a really important co-occurring condition in people with dementia, and particularly around the time of dementia diagnosis. And providers should work to provide care for people with dementia to address these mental health concerns. And in a similar snapshot, we also examined the relationship between dementia, previous dementia, and the rate of being diagnosed with a stroke. And similarly, we found that there was an increased rate of stroke in the shorter time period following a dementia diagnosis. And this indicates that stroke and dementia likely have shared risk factors like cardiovascular risk factors, and patients should be managed in order to control these risk factors to reduce the risk of further health outcomes. Now, in our third research snapshot, we looked at sex differences in the use of antipsychotic and benzodiazepine medications. And these are medications that are often used off-label to treat the neuropsychiatric and behavioral symptoms of dementia. And we looked at these in a long-term care home population. So we know that the use of these medications is common in people with dementia, but it's not without risks. Previous studies have shown that these medications have been associated with poor health outcomes like falls, stroke, pneumonia, increased cognitive decline, and mortality. And these medications are sometimes known as potentially inappropriate medications because clinical guidelines often recommend not using these in people with dementia for the reasons that I've mentioned. So we know that men and women who have dementia can have differences in their psychosocial symptoms of dementia. Men may be more likely to exhibit challenging behaviors, and women may be more likely to have anxiety and depression, for instance. So we wanted to investigate whether the patterns of the use of these medications was different between men and women. So overall, we found when we looked at people who were newly admitted to a long-term care home who had dementia, we found that men were about 27% more likely than women to start an antipsychotic in the 180 days following long-term care admission, while women were less likely to stop an antipsychotic, and they were also less likely to stop a benzodiazepine medication. So overall, women were less likely to experience discontinuation or stopping of these medications. So overall, these findings indicate that antipsychotics and benzodiazepines are being used differently to treat the symptoms of dementia in men and women. And as we know the use of these medications can be risky, it's important to further understand factors behind their use and to avoid their use where possible. So what do we take from this in terms of how does our research inform what we can understand about population health for people with dementia? Well, to summarize, in our COVID-19 work, we showed that there was a large decline in health services for people living with dementia in Ontario during the pandemic. And this observed decline can have negative impacts in terms of the future long-term health outcomes for people with dementia. And this is important to assess in future work. For our dementia and psychiatric comorbidity study, we showed that people with dementia were at an increased risk of developing other disorders like mood and anxiety disorder and stroke. And it's important to recognize that these conditions can be treated and can help to improve health outcomes with appropriate management and health system planning. And finally, for our sex differences study, we found that men and women who were newly admitted to a long-term care home experienced different patterns of starting and stopping medications. And these medications should be regularly reviewed and acknowledge that there may be potential sex differences in their use. So coming back to the idea of HANDS Ontario, so how will the ICS or health administrative data be used? Well, it's going to be talked about next in greater detail, but we'll give you a bit of a snapshot here. So data from the HANDS Ontario participants who are shown here in orange will be linked using those anonymized health identifiers that Susan alluded to better understand health services use and neurodegenerative diseases and to look for long-term outcomes in this patient population. So by looking at the health administrative data, we can understand what someone's comorbidities or prior healthcare use was prior to their dementia, and we can also follow them longer term to understand how they interact with the healthcare system and whether, for instance, they're admitted to a long-term care home. So we'd like to thank you so much for attending today, and we're happy to answer any questions that you may have, or feel free to reach out to us at the email addresses provided. I know we are a little bit short on time, but we'll just leave you with a few questions to mull, and you can write in the chat box if you're able to. We're curious about your experience during the COVID-19 pandemic. Have you used virtual care? What was your experience? And any other suggestions you might have for us. So thank you so much.
And feel free to reach out to us outside of the Zoom fora too, if you have other thoughtful answers to these questions too.
Absolutely.
Thank you all very much.
Absolutely.
Thank you very much. I really appreciate it. That was terrific. Perhaps you can read the introduction to Dr. Swartz.
Yeah, sure. I'm happy to introduce Rick. So Dr. Swartz completed his training and his neurology residency at the University of Toronto, and his appointments and affiliations include being a stroke neurologist at Sunnybrook Health Sciences Center, an associate professor in the Division of Neurology, Department of Medicine at U of T, and directing the Sunnybrook Stroke Research Unit. He's also the medical director of the Northeast GTA Regional Stroke Network and the director of the University of Toronto Stroke Program, and also the co-lead of the Andre study. And as a clinician scientist, his clinical specialty is stroke neurology with a focus on stroke in the young, stroke in pregnancy, intracranial vascular diseases, and the functional consequences of stroke, including vascular cognitive impairment. His research focuses on improving functional impairment in stroke, from hyperacute clinical trials to long-term complications, with a focus on intracranial vascular diseases, including small vessel disease, and the cognitive impact of stroke and neuroimaging biomarkers of intracranial arterial disease. So welcome, Rick, and go right ahead.
Thank you very much, and thank you to those of you who have attended all morning. It's been an amazing morning. I apologize, I've had to be a little bit in and out. I'm in the hospital on stroke call today, so hopefully my pager doesn't go off in the next few minutes. Well, we kind of bring this home, showing you a little bit about HANDS Ontario and the study itself, and really hopefully bringing together a lot of the science and background that you've heard this morning as to why we're doing this and what its potential impact may be. So the Hands Ontario study is looking at health in aging, neurodegenerative diseases, and dementias in Ontario. And we use the hand metaphor in part because there's a number of worldwide studies that use that part of the anatomy. Several years ago, there was a finger study that looked at vascular risk reduction and exercise and activity in reducing dementia. And since that time, there's now a worldwide fingers effort. There's the CAN Thumbs Up study, and the hands study is really fitting into that analogy, but it also is an apt description of what we're doing. We are part of ONDRI, which you've heard about today. And just to remind you from flashing back to Mario's introductions this morning, really this is what we're for. What all of the scientists and physicians that you've heard from today are devoted to improving the diagnosis, prognosis, care, and outcomes for people living with neurodegenerative diseases, stroke, and dementia. And we're really looking at the cross-disease, platform commonality. So looking at not just what drives vascular cognitive impairment or cognitive impairment after stroke, but what may contribute to, as we heard about, those co-occurring, those comorbidities, as you just heard about here, and again, what Dr. Masalis spoke about earlier this morning, the overlap between Alzheimer's and stroke, or FTD, or Parkinson's and Alzheimer's, or ALS and FTD. All these diseases interact, and our categories, we think of these as discrete boxes, but they're really much messier than that. And really, the initial foundational study that looked at all these biomarkers that Dr. Munoz was talking about has provided a foundation that we can build on in Hands Ontario. And Hands is really designed to build on what you've heard: remote sensor technology, wearable devices, genetic and molecular biomarkers, to really help us make discoveries on multiple levels across the basic science, individual function, and health system levels. And you've heard a little bit about all of those today. And Hands is really designed to try to understand how these different diseases affect people, how they may interact and overlap, how we might be able to get markers to help us diagnose better, but also to help us individualize management. And Dr. Masalis was alluding to that kind of complexity and the need to make things individual. I think you saw that also in Dr. McElroy's presentation when he was showing just the walking patterns for different people. We really need to target individual health behaviors, but potentially in the future, individual therapies. And Hands is really designed to look at all those different levels of what we're intervening on, how we can intervene to make somebody's day-to-day activity better, and how we might be able to use the health system and use interventions to improve outcomes for populations. So what does Hands really mean for a person? What are we asking people to do? It's designed, and I want to emphasize that this was designed before the COVID-19 pandemic. And before equity, diversity, and inclusivity, EDI was a whole big thing over the last few years, and importantly so. But it was designed with those ideas. It was designed to get out of the lab, to get to the community, to get to the home, to get to where people are living with these diseases. And it was designed to be more accessible geographically, socioeconomically, for people who may not be able to get into large academic centers or take large amounts of time off of work, because it's designed to be done from your home with remote assessments, telephone calls or video calls, mail out, mail back samples. There's one in-person visit that's a lab test, which I'll show you in a second, but it starts with a screening and informed consent. So we do this by phone or by computer, and our coordinators call people who express interest to their doctors or off our websites or after talks like this, or are referred from friends or family. And they'll go into detail about the study. There are multiple study visits spread out over time, so you don't have to get worn out at any one visit. And it's about an 8-week arc, as Dr. McElroy was telling you earlier. We mail out sensor packets, the 3 sensors, the wrist, the chest, and the ankle that he was speaking about. And that's worn for 7 to 10 days. And it's sort of a 10-day period. And there is that opportunity to take it off if you're, well, I don't know, if Omicron settles down and we're ever going out for nice dinners again, and you want to not have to be wearing sensors for an evening, there's that opportunity to disconnect for an evening as part of that 7 or 10, that 10-day cycle. But we're looking for at least 7 days of continuous wear. And the blood sample can be given at any Life Labs anywhere in Ontario. And so it's quite accessible, doesn't have to come into an academic center, don't have to attend at a hospital. You can make an appointment again at your own convenience. And then completing the assessments and questionnaires by phone and online. Pardon me, near the end of the study, as you saw from Dr. McElroy, we're producing personalized health and activity reports with the Health Canada approved outputs from these devices so that we have evidence behind them. And then we're seeking our volunteers' feedback on how to make the information that they get the most useful and most productive it can be. The feedback forms that we're giving are already tremendously informed by almost 100 volunteers already, and by our Patient and Community Advisory Committee, our PCAC, to be hopefully useful now. And we want to continue to iterate that to make it more and more useful, and maybe even work towards giving feedback forms for physicians that can help with management. So this is really a partnership with our PCAC. And the Patient Community Advisory Committee has been instrumental in really developing this study in general and in launching it. And one of the people that we have with us today, Mr. Larry Linton, has been a very active member of our PCAC, our Patient Community Advisory Committee. I'm going to introduce him now, and we'll ask him to say a few words. Larry is an immigration lawyer by trade, having been trained in his native South Africa and then reestablishing himself in Canada following his own immigration. He was diagnosed with Parkinson's in 2012 and found the first 2 years after his diagnosis very challenging. He felt withdrawn from his family and found it easier to deny what he had. But after an existing initial phase of denial, Larry found that some acceptance with his diagnosis. And with that acceptance, he literally got off the couch and reclaimed his life, which has, he says, in many respects become more rewarding, fulfilling, and meaningful than at any time before his diagnosis. He added author and musician to his long list of achievements after his diagnosis, and is currently the chair of Parkinson's Canada's Parkinson's Advisory Council. And as I mentioned, part of Andre's Patient and Community Advisory Committee, advocating not just for persons with Parkinson's disease, but across the neurodegenerative and stroke spectrum. By sharing his story, Larry hopes to inspire others to live their most authentic and best lives with Parkinson's and neurodegenerative diseases as well. And we're thrilled to have him here. So thank you, Larry, for joining us.
Well, thanks, Dr. Schwartz, for that introduction. I just want to make sure that you can all hear me. Great. The day of my diagnosis 10 years ago was the start of a very dark chapter that saw me withdraw from my family, my friends, my work colleagues, until I was living on the couch, literally. But 2 years later, after going through the other stages of grief, I finally reached the point of acceptance and started living with the disease and no longer against it. I started to reclaim my life, to reinvent myself, and to regenerate a life that was worth living again. I returned to my love for drumming. I started running and reached the professional milestone of partner admission 4 years after my diagnosis. But I've come to realize that it's just as important to look to the future. COVID has forced us all to do the same. Our lives have fundamentally changed over the past 2 years. We live differently, we work differently. And it has made us look into the future too. Before COVID, I used to arrange my follow-up appointments with my specialist in the early mornings when I was on, both physically and mentally. I was hoping to show him how well I was doing, that I was beating this thing called Parkinson's. For the short time that we had together, I didn't mention the subtle changes that I was becoming aware of. Instead, I highlighted the long hours I was still working and downplayed some cognitive symptoms that I was starting to experience. But with COVID, the delivery of healthcare has changed. Telehealth has brought healthcare delivery into the home environment by connecting us with our medical professionals via Zoom, just like we are doing today. Instead of relying on historical information and home diaries, telemedicine has allowed us to engage with our professionals in our own environment in real time. putting us at the center of our own care and management. Rather than focusing on our concerns, this approach considers us, the patient, as a human being in a social and geographic environment. For me, it has essentially invited those in my circle of care into my daily life to see me in a more authentic and genuine state, and has allowed me to be vulnerable in dealing with the progression of the disease. Andre's mission of embedding research into care and care into research is another important step into the future. And having been a participant in the HANDS Ontario study is another great example of how care can and will be delivered in the future. As you've all heard today, the study has three main components, looking for blood-based biomarkers, symptoms and behavior tracking and reporting through wearables, and the long-term disease trajectory. Over the course of 10 days, I wore the chest monitor and wrist and ankle sensor. I did experience some difficulty with attaching the chest monitor. I had trouble peeling the adhesive tape off. It's a bit of a challenge with tremors in your dominant hand. So I'd recommend that the adhesive tape maybe be wider. It would be easier to hold onto and strip away. The sensors in combination track my sedentary behavior, my physical activity, my sleep patterns, and nighttime behavior. Unfortunately, I lost the ankle sensor somewhere on a 100-kilometer bike ride, but I promise you I completed the ride, but I couldn't find the sensor. I found the devices generally easy to wear. My one recommendation is to make them all waterproof so that they can be worn continuously throughout the monitoring period. I was satisfied with the overall results that came through a review of a personalized health activity report and found my sleep patterns and nighttime behavior quite revealing. I now know why my wife sometimes looks as exhausted as I do in the mornings. The research team was great. They were engaging, accessible, and helpful, even remotely. The feedback sessions were informative and helpful. They really care about everybody's experience and the feedback and are working hard to make the research better and better. As a person living with Parkinson's, obviously the first prize is to find a cure and to talk about having had Parkinson's. But until then, the next best thing is to be able to live with it and maintain a good quality of life for as long as possible. We live with the disease every day, every hour. Efforts like the HANDS Ontario study go a long way to empowering us to take control and be an active participant in the management of the disease instead of being a passive recipient. I'm thankful for having had the opportunity to participate, and my hand is firmly up in the air to participate again. So thank you very much.
Thank you, Larry. That's tremendous. We really appreciate both your participation in HANDS, your participation in the PCAC, and your willingness to share your experiences with us today. We just so very grateful for your collaboration. And I think with that, we'll leave the chat open and have the last couple of minutes with questions. I'm going to emphasize that HANDS Ontario is open to adults living independently with no diagnosis. So oftentimes that's partners and family members of participants. Some couples are enrolling as a family activity. Individuals with MCI, mild cognitive impairment, Alzheimer's, Parkinson's, ALS, frontotemporal dementia, stroke. It's a broadly inclusive study. And I'm going to leave the contact information up here, cell phone numbers, emails. You can check out the study on our website. And I just want to thank all of our speakers today for a really engaging and hopefully interesting and valuable day. So to Dr. Masellis, to Dr. Munoz, to Dr. McElroy, to Drs. Bronskill and McLaughlin, and to Larry for joining us. Thank you very much, and thank you all for your attendance today. This is we're really excited to have this. For those who want to, it will be recorded and available, and we'll make some of the snippets also available more publicly, I suspect. And if anybody has questions or comments, we're happy to keep the speakers on the line. Thank you again, everybody.